expression of markers linked to neuroinflammation, TNF-alpha, IL-1 beta, and IL-6. We find some changes, but we need to further investigate these changes. So then we sent some samples for TempoSeq. And when the samples we sent were after the acute exposure of 48 hours and after the repeated exposure of 1 week. And what we could see after performing DSEC2 analysis is that there's differentially expressed genes in all the conditions. And interestingly, there's more differentially expressed genes after the acute exposure than after the repeated exposure of 1 week. These genes are linked to different biological processes such as neuronal differentiation and development, and also lipid metabolism that is quite consistent in all the conditions that we tested for. If you look here to this graph of the 2 micromolar of amiodarone exposure at 48 hours and after 1 week of exposure, we see that these genes are linked, such, for example, to regulation of lipid metabolic process and regulation of steroid metabolic process. So this data gave us an indication that amiodarone can be affecting the lipid metabolism in the brain. And we already had indication that amiodarone is affecting the lipid metabolism in other organs, such as the liver, where when a lot of patients that take amiodarone for a long time end up by having steatosis, that is the fat liver disease. So we checked for some other markers for lipid metabolism and in gene expression, and we see that the novel lipogenesis seems to be affected here with the FASN expression, and also the formation of lipid droplets with PLIN2 and PLIN4 expression being affected. So in general, the distribution kinetics show us that amiodarone is accumulating inside the cell over time. There's no detection of the main metabolite of amiodarone, what indicates us that any effect that we see is probably being made by amiodarone itself and not its metabolite. We see that astrocytes are early and strongly affected by amiodarone. Neurons are affected later, so after the repeated exposure, particularly GABAergic and cholinergic neurons. The lipid metabolism may be impaired, and since astrocytes play numerous roles to sustain neuronal function, their impairment may indirectly trigger neuronal stress, not just the effect of direct effect of amiodarone. This data will be used for the in vitro to in vivo extrapolation in vivo that Susanna is working on, and she will also use data coming from Sarah that is working with the BBB. Then I will now show you data coming from another chemical out of the 10 from the list of IN3, Paraquat. Paraquat is a widely used herbicide. We cannot use it anymore in Europe, but in the United States it's still possible to use, although there's a lot of restrictions, and it's still widely used in development countries. We know that it's toxic and it is a health and environmental hazard, affects the redox cycle and mitochondrial function, it's produced developmental neurotoxicity, eye damage, Paraquat tan, mucosal injury, pulmonary fibrosis, heart, liver, and renal failure. And me, Pranika, Zara, and Cormac, we took advantage of the fact that we have all these models representing different organs that you can see here that were exposed to Paraquat, and samples were sent to TempoSeq as an opportunity to try to understand if the response of all these organs is in the same direction, the response to Paraquat is in the same direction or not. So with the data coming from the TempoSeq, we can extract the differentially expressed genes, the pathways that these genes are linked to, and the potential transcription factors being affected by these differentially expressed genes in the data coming from the TempoSeq from these different models, to try to understand what could be the public and the private responses to Paraquat per organ. So when we plot the different controls and treated samples of the different models, we see that they cluster together based on the cell type they are, so podocytes, hepatocytes, endothelial cells, brain spheres, neurons, BBB cells, and PTL. Looking to the differentially expressed genes in at least 2 of these models, we end up with a lot of pathways being potentially affected, of course, and such as some related to P53, NRF2, ATF4, and also ESR-mediated signaling. That is actually the only pathway common to all the models that is related to estrogen. And even when we look to the transcription factors that could be affected using this Viper package in R, we see already also some transcription factors coming through as potentially affected that are linked to these estrogen and androgen pathways. And indeed, when we look to some papers, we do some bibliography research, we already see estrogen being affected by Paraquat in the brain. So we are quite enthusiastic about these results. Finally, along all the last 3 years, I've been trying to add microglia to the brain spheres. And why? Because microglia, they are the resident macrophages of the brain. They are the cell type that is missing in our model. And they are very important since they are involved in a lot of developmental process of the brain, but also on keeping the homeostasis in the brain. And we are very interested in inflammation, and they are one of the key players in the inflammation. So we are very interested on having these cells together with our brain spheres. So I've been trying this since the beginning, and I've been having help of different students, master students, Lorena, Tanya, Kelvin, and Karina have been helping me. Here I'm showing what we did when Kelvin was with us. We were receiving microglia progenitors derived from human-induced pluripotent stem cells by Sally Cooley in Oxford. And we were trying to add these progenitors to our neuroprogenitor cells and produce the brain spheres. And we were able to have the cells inside. So in red here, you see the microglia progenitors inside our spheres, and where we could see a very nice morphology. And then this morphology would change when we will expose the cells to LPS and interferon gamma to a more reactive morphology of microglia. So these cells could indeed respond to this LPS interferon gamma. So we are very happy to being able to do this, but we need to further work on this model. So I would like to thank you for listening to me for this while I was talking about my project. I would like to thank my lab and particularly Gabrielle Zurich for giving me the opportunity to work in this project from the beginning. And I would like also to thank all the In3 family. It has been very nice to work with all of you, and it has been a privilege to be part of this very nice team. So if you have any questions for me, you can ask them in the chat, but also if you can send me an email or find me in LinkedIn. Thank you very much. Enjoy the rest of the videos and have a nice day.